As can be seen from the graph, this is the concentration of ethanol. This is the concentration of methanol. So when presented with both carbon sources at once, ethanol is consumed first, and then it switches over to produce enzymes to metabolize methanol. And here's a lag time showing the first growth phase for ethanol, then it kind of stabilizes, and then the second growth for utilizing methanol. So with these metabolism pathways, ethanol is preferred over methanol. When we was looking into this metabolic pathway, we noticed, or we found that there's a major enzyme called alcohol oxidase. It is the first key enzyme that's present in this pathway. It just basically metabolizes or converts methanol into formaldehyde. There are two related alcohol oxidase enzymes that are produced by two separate genes, AOX1 and AOX2. However, when we were researching both of these genes, we found that the mRNA transcribed from AOX1 has a higher steady state level. In other words, it's degraded less quickly as compared to that produced by AOX2. And in turn is why there's the majority of this enzyme is traced back to the AOX1 gene. So therefore, we went along with using the AOX1 gene for this project. Our goal was first to just combine this promoter, the AOX1 promoter, with something that can allow us to detect when this promoter is induced. And so we just used an RFP gene, a red fluorescent protein gene. We wanted to place this system into E. coli and see if it could operate as we wanted it to. And then we would do further testings. So our first step for the construction of this part was to first just isolate this promoter from the AOX1 gene. I believe we tried to obtain some cultures of Pichia pastoris by ourselves and tried to remove or separate this promoter independently. However, this wasn't working too well. But we fortunately found a professor from University of Chicago, Dr. Ben Glick, that had already isolated this AOX1 gene, and he sent us this gene in the form of the PIC3K vector. So our first goal was to see if there were any of the 4 standard BioBrick enzyme restriction enzyme parts or sites within this promoter. Fortunately, we found none, so that made it a little bit easier. So then we just tried to remove this promoter from, we didn't want the entire gene, we just wanted to use the promoter. So we amplified this promoter using PCR. And to kill 2 birds with 1 stone, we basically, in designing the primers using PCR, we attached the suffix and prefix standardized restriction enzymes. So once the PCR was completed, it created a product that had the restriction enzyme sites already on them. We then cloned this product into another vector, the PCR 2.1 vector. This was purely to make it easier to amplify this promoter, so we wouldn't have to continuously do PCR just to continuously get more copies of this promoter. In this vector, it already had a EcoR1 site in it as well, which we used to our advantage when trying to confirm that it was the exact product we wanted. We then took this vector and transformed them into E. coli cells. And from the screen, you can tell that there are 2 different types of colonies, one blue, some white. Within the PCR 2.1 vector, there is a LAC-Z gene. And normally functioning, LAC-Z gene produces beta-galactosidase, which just basically cleaves X-gal. And these products produce a blue pigment. However, in this vector, the insertion point for new DNA disrupts this LAC-Z gene. Therefore, if there was new DNA inserted, there would be no beta-galactosidase produced, and therefore no blue pigment. Therefore, the colonies that are white are the ones that contain this vector in our promoter. Just to confirm that this is the the part that we had the part that we wanted. We just took advantage of the two EcoR1 sites and did a restriction enzyme digest and ran them through a gel electrophoresis. The blue circle represents, the band in the blue circle represents the remaining vector without its promoter, and the yellow band represents the promoter itself. Our next step was then to prepare the red fluorescent protein vector in order for ligating it with our promoter. And we obtained this vector from the iGEM registry, and we just transformed this into E. coli cells just to amplify them. And as you can tell, they exhibit a very red color as well as fluorescence under UV light. We then, using extraction kits, removed these vectors from the E. coli cells themselves, E. coli cells, and prepared them to be ligated. In this preparation, in order to confirm once again that this was the actual product that we wanted, we did restriction enzyme site restriction digest using EXPO1 and SPE1 sites. This removed both the promoters of these vectors, and here again we ran another gel. The blue again representing the remaining site from our part, and the promoter itself represents this. The red encircled is the band that representing the vector with our RFP, and the promoter that came with this RFP vector was too small to be detected on this particular gel. We then performed the ligation of our promoter with this RFP vector. We did this overnight at 16 degrees Celsius, and then transformed them into competent E. coli cells. And I don't know if you can see, but there are cells, some cells which are white and some cells which are red. So our current research was that we're working, the current stage of research that we're at right now is trying to determine which of these cells are the ones that contain our ligated product, if any. We're having problems with this because all the ones that we've screened, we have not found that they have both the AOXI product coupled with the RFP. This could be due to a number of reasons, certain enzymes not working or inefficient ligation processes. So we're trying to determine this by, we're trying to improve our ligation method using controls and purifications. But as of now, we have submitted our particular part, the AOXI with its standard restriction enzyme sites to the iGEM registry, so we have submitted that part. So we haven't yet developed the system of the AOXI coupled with the RFP, but in theory, once this is done, we can determine if E. coli can, in fact, detect the presence of methanol and ethanol and the amounts of it. Basically, there would be a direct measurement to determine the amount of methanol. If we can just introduce methanol to the system, the methanol will be consumed and the cell will begin to will turn red and fluoresce under UV light. And using this lithium time to determine, we can use this lithium time to determine how much methanol is present. However, it's more of an indirect way to determine how much of ethanol is present, because at first, since ethanol is preferred, it will be consumed first. And then once all of the ethanol is consumed, methanol will be consumed. Once methanol is consumed, our AOXI promoter will be induced, which will express the RFP gene. So we would use the time, so in order to determine the amount of methanol present, we would use the time before the cells fluoresce. So this is just a general overview. Introduce the system to both ethanol and methanol, time until it fluoresces or before it fluoresces, and then relate this time to the amount of alcohol that's present. The ligation, the obstacles that we have encountered throughout this project have, again, as stated before, we have not found cells that had our ligated product of AOXI and RFP. So we're trying to determine what exactly went wrong, if there were no restriction enzymes, or there were no enzymes that we had, I guess, inefficient enzyme restriction digest, or if there was just the ligation did not work. So, and we're also trying to perfect our screening and control methods. Further or future obstacles that may arise would be the functionality of the part in E. coli. Since we don't know that this same metabolic pathway of ethanol and methanol will be present in E. coli, we may have to just put this system back into Pichia pastoris yeast, or maybe perhaps into a similar yeast, which we're looking at Saccharomyces cerevisiae. Again, there is, as stated before, there is an indirect measurement on ethanol. So, and this has to deal with the response and degradation and lag time. It takes a certain amount of time for cells to switch over to produce methanol, so we cannot take the entire length of time to and relate that to the amount of ethanol that's present.
We have thought about the potential problems that we will have if we are able to create this part and have it working. So we are playing with some ideas for mathematical modeling, which can be a pretty important tool. We haven't started any of it yet, but some of the ideas we are thinking about is determining the minimum concentration of ethanol needed to start this process and the maximum concentration of methanol. And as Corey said before, the lag time, as far as protein synthesis and folding rates, and the rate of degradation, so we can get a better idea of actually how to relate how long it's fluorescing and how much alcohol is in the system. We also need to determine the minimum amount of enzyme that's needed before reaching detectable levels of fluorescence, and how this would ever be implemented in a real industrial process. I believe one of the most important things of iGEM is what it is that the students gain from participating in this. And we all gain many skills and techniques, both in the lab and out of the classroom. As far as collaboration and teamwork, it takes everybody to get this going. And a few of us know that it's also it looks good to potential employers, that's for sure, when they see that we're able to come together and work on these projects. A lot of people don't have any idea what it is that we're doing. And it's usually pretty impressive to most employers. And we've also been able to create some ties with companies such as Monsanto and Midsize Scientific, who are donating money and getting to know about the university and what the students are doing. I'd like to give special thanks to Dr. Haiti Shannon and Dr. Dave Westenberg for all their guidance and support through this project, as well as Dr. Ben Glick from the University of Chicago for his generous gifts, and for the following funding sources for helping us come to the iGEM competition. Thank you for your attention, and before we open it for questions, I'd like to ask my teammates to come up.
Are there any questions? You guys want this?
So you mentioned the indirect measurement of ethanol. Have you guys thought about ways that you might begin to measure that directly?
So basically you wanted to see if there was a method to directly measure ethanol. In our research, when looking for this yeast, they know very little about these metabolic pathways of methanol and ethanol. We did try and look for more to see if there was an isolated gene that expressed a certain enzyme to metabolize ethanol, but from our knowledge, we had not found one. And so we ran across the one for methanol, so okay, we can just use this one. But yeah, in efforts, it would be easier to use something more direct because we wouldn't have that whole lag time involved. But like I said, there's not much known about these processes.
Okay, thanks for the presentation. I think it's a really interesting idea. I was wondering, go back to the beginning when you were thinking about this idea, why you decided to take a system that's already functioning in Pichia pastoris and try and put it into E. coli instead of just putting the detection mechanism into the Pichia and then doing the fluorescence in that organism.
I believe we wanted to First of all, it was more or less a fundamental project. Once we had all came together, we had people from various majors. There were some biology, some chemical engineering, some I think we had some electrical engineering as well. So it was more of like a fundamental process, just so everybody can understand like the basics of this project and in biology. We also wanted, and go back to this picture, with the toxic growth, there was. They tried, in this model, they used like small concentrations of ethanol present, and it took 20 hours for it to consume. And I mean, when we initially wanted to try and make this into a breathalyzer, you know, it wasn't very efficient to wait 20 hours to tell if you were inebriated or not. So we were trying to look for a faster method. However, when doing this, we found that, okay, well, it may not exactly work. We haven't actually created the part to see if it will be functional in E. coli, but if so, if we could possibly make things move along a little bit quicker. We also thought of ways to, I guess, to make it more quicker to detect it, such as using bioluminescence, so that, you know, it's, once you see it glow or something, then it's quicker than waiting for the cells to turn red and fluoresce. But does that answer your question?
Besides, outside of the breathalyzer idea, where do you see this kind of project going forward? Like once you have a bacterium that, yeah, you know, detects ethanol and methanol and it works like that, where do you see this being put?
You want to take?
Well, with like home brewing, if if there's methanol present, you might go blind. So maybe a bacteria would be introduced, and that'd be a quick way to tell.
It's in many cases it's hard to tell, you know, if it's really, you know, industrially feasible, because there are, you know, much quicker, you know, breathalyzers that you don't need to wait so long. But it was more of a goal to see if we could do this or not. And so we were trying to just see it, and also to get the other people that weren't as aware of biology and had much knowledge about it, just to see what's capable. So.
Okay, great. Thank you.
Thank you.